Let's continue our discussion on the nephron. We see the nephron here, and in pink on the left, we see the glomerulus with the afferent arteriole entering the glomerulus and efferent arteriole leaving the glomerulus. To be clear, this region right here, the renal corpuscle, which is the combination of the glomerular capillary and Bowman's capsule, that makes up the renal corpuscle, and that's where filtration occurs. And that's what I want to focus on with this particular lecture. That is to say, what gets filtered, what doesn't, and how is that regulated. In this image right here, we see an expanded view of the renal corpuscle with the afferent arteriole coming in to the glomerulus, efferent arteriole leaving the glomerulus. This is a peritubular capillary. This is the beginning of the proximal convoluted tubule. The proximal convoluted tubule and the majority of the downstream nephron. And when I say that, the proximal convoluted tubule, distal convoluted tubule, and portions of the loop of Henle are lined by simple cuboidal epithelial cells that we see right here. Once again, the nucleus is roughly right in the center of those cells and fairly circular in shape. The space between the two simple cuboidal layers is the lumen. This right here is the capsular space. And this space is where filtrate first arrives to. So filtrate's going to move from the glomerular capillary into the capsular space. Now, Bowman's capsule is composed of two layers, a parietal layer on the outside of it and a visceral layer on the inside. Parietal layer and visceral layer. The visceral layer does not exactly look like this, and I'll show you a different version of this image in a moment. But I just want to highlight that the parietal and visceral layers of Bowman's capsule are composed basically of simple squamous epithelial tissue. That's certainly the case for the parietal layer. The visceral layer has specialized epithelial cells known as podocytes, and we'll talk about those extensively. So capsular space, parietal layer, and visceral layer of Bowman's capsule. Quick review, capillaries, and once again, the glomerulus is a capillary bed, is composed of endothelium, simple squamous epithelial tissue. So we have simple squamous epithelial tissue and a very similar form of simple squamous epithelial tissue of the visceral layer. These two layers are abutting each other. That is to say, they are right next to each other. The only thing separating these two layers is a basement membrane, an intervening basement membrane, really a basement membrane for both of these layers. Keep in mind that all epithelial tissue sits on a basement membrane, a layer of connective tissue. So if substances were moving from the capillary bed into the capsular space, it would be crossing the simple squamous epithelial cells of the glomerulus through its basement membrane, through the basement membrane of the visceral layer, and then through these epithelial cells that we refer to as podocytes. We have previously talked about different types of capillaries, and one was a type of capillary known as a fenestrated capillaries that have pores in them. And all these black dots are representing pores or fenestrations that will allow entities to leave the blood plasma and to enter the capsular space, becoming what we know as filtrate. So I want to say there are two mechanisms that prevent unwanted things from being filtered. And when I say unwanted things, we have discussed that everything from the blood gets filtered with the exception of the formed elements and most plasma proteins. So red blood cells should not be filtered. White blood cells should not be filtered. Platelets should not be filtered. And most plasma proteins should not be filtered. And that is prevented by the size of these molecules. That is to say, the formed elements, by and large, are too great in size to fit through the pores, or some other avenues that we'll talk about related to the podocytes. And additionally, the basement membrane, which we can't see here, but that intervenes between the endothelium of the capillary and the epithelium of the visceral layer, that basement membrane has a negative charge. Most plasma proteins have a negative charge. So if plasma proteins are small enough to squeak through these fenestrations, hopefully the negative charge of the basement membrane is going to repel those plasma proteins. And theoretically, that is what's going to happen. Keep in mind that like charges are attracted to each other. Say a sodium ion, which is positively charged, and a chloride ion, which is negatively charged, they are attracted to each other. But like charges are repelled from each other. So if a plasma protein gets through the pores but encounters the negative charge, they're not going to proceed through this filtration membrane. And I was remiss to even use that term until now. What we are talking about is the filtration membrane, which is the combination of the glomerular epithelial tissue and the epithelial tissue of the visceral layer of Bowman's capsule. And this really is what we're looking at right here. The combination of these two layers, the endothelium and the visceral layer, make up the filtration membrane. Now, the diameter of these capillary pores is roughly 10 nanometers. It could be as large as 80 nanometers. The numbers are highly variable because every article I read on it or textbook, there's different numbers. The permeability across the fenestrations of the glomeruli is much greater than in other capillaries throughout the body. But the diameter of the pores, let's say, is in the realm of 30 nanometers. Now, that is a very, very, very small diameter. To put that in perspective, red blood cells are roughly 7.5 micrometers. White blood cells are anywhere from 1 to 17 micrometers, and platelets are approximately 3 micrometers. Those are too large to get through the capillary pores. Now, one thing that may be confusing is I told you the capillary pores are roughly 30 nanometers in diameter, and I just described the red blood cells, white blood cells, and platelets using the unit micrometer. A micrometer is much larger than a nanometer. So let's take a quick look at the relationship between different units of measure. I don't think it's important to memorize what I have written down right here, but it is important to understand the relationship between these different units of measurement. So in this first line, this is representing a meter. A millimeter is 1/1000th of a meter, and that would be represented just by this yellow vertical line right here. This is 1/1000th of this entire line right here. So a millimeter is 1/1000th of a meter. This entire yellow line represents that 1 millimeter. That is to say, this entire yellow line is represented right here on the above vertical line. 1/1000th of a millimeter is a micrometer. 1/1000th of a millimeter is a micrometer. So same idea here. This whole yellow line is a millimeter, but this portion of it, just the green portion, represents a micrometer. Then we have the micrometer right here. Keep in mind this symbol that looks somewhat like a U represents micro. So this is a micrometer. 1/1000th of a micrometer is a nanometer right here. So the point in all of this is that a nanometer is much, much, much smaller than a micrometer. 1 micrometer is equivalent to 1,000 nanometers. So when we talk about red blood cells being only 7.5 micrometers, white blood cells within the realm of 5 to 17 micrometers, and those numbers I gave you previously, those are extremely large compared to the size of the fenestrations or pores in this capillary bed. As a result, that's why they do not get filtered. Some plasma proteins, once again, can squeak through these fenestrations. Hopefully, the negative charge of the plasma membrane repels them. But there's another mechanism based on size to help repel even more. And one thing I do want to say is that basement membrane is also somewhat porous, but it only allows things that are smaller than 8 nanometers. So just to put that in perspective, if I say a red blood cell is 7.5 micrometers, that means it's going to be 7,500 nanometers, which is dramatically larger than what is permitted to pass through the basement membrane. So like I said, this visceral layer doesn't really look like this. So we're going to take a look at a different representation of this to get a better perspective. Okay, so here we have the visceral layer, and the specialized epithelial cells are known as podocytes. So each one of these, we can see the nuclei in the center of these are the podocytes. The podocytes have these cytoplasmic extensions that radiate out from the main cell body. And coming off these cytoplasmic extensions, we can see what are known as foot processes. So if we take a closer look at this, these are all foot processes right here. And between the foot processes are what are known as filtration slits. And those are another gap or space that substances need to squeeze through to make it into the capsular space, to make it into the filtrate. These filtration slits are roughly 30 nanometers wide. So they are inhibiting the white blood cells, red blood cells, and platelets from moving through here. There are going to be some very small plasma proteins that can make it all the way through the filtration membrane, but by and large, most plasma proteins are not permitted to pass through. So the filtration membrane works by two mechanisms: by inhibiting larger molecules from passing through the fenestrations or pores of the glomerular capillary. Also, the basement membrane inhibits molecules based on size, as do these filtration slits. So there's 3 mechanisms that are inhibiting molecules based on size. And the 4th mechanism is the charge of the basement membrane, the negative charge, which is going to repel the negatively charged plasma proteins. And when I say plasma proteins, I'm talking albumin, fibrinogen, plasminogen, antibodies, what have you. So then what gets through here to make it into the filtrate? It's everything else that we find in the blood. It is water. It is nutrients, fatty acids, amino acids, glucose, hormones, medications like insulin get through the filtration membrane. Once again, 99% of the filtrate, 99% of the blood plasma that gets filtered is ultimately going to be reabsorbed. But this is how things get filtered across the filtration membrane, which is the combination of the endothelium of the glomerular capillary and the epithelial cells of the visceral layer of Bowman's capsule.